Okay, in this video, what I'd like to do is cover the material we did in class yesterday, and this material is going to be about lipid management. And what I mean about lipid management is going beyond what we talked about before about fatty acid synthesis and the uptake and assimilation of dietary fats in the body. And this is how the fatty acids and cholesterol are actually transported through the body and how they become useful to the tissues. So let's start out by talking dietary fats. So here's a representation of our intestine, and we have dietary fats that need to be taken up by the body from the lumen of the intestine. These dietary fats are digested, as we have seen, by our pancreatic lipase, which is going to take our dietary triglycerides and convert these into 2 fatty acids plus our 2 monoglyceride structure, and then these can be taken up by our intestinal epithelial cells. And also recall that the fatty acids are primarily being taken up by the fatty acid transport protein. Now, the other part of our dietary fats are cholesterols plus cholesterol esters. Now remember, cholesterol is just the base structure, and cholesterol can be taken up by our NPC1L1 transporter. This is a cholesterol transporter. And this is located on the apical surface of the intestinal epithelial cells, and it's involved in taking cholesterol up from the lumen and into cells. The cholesterol esters, however, have to be processed, and these are processed by esterases, cholesterol esterases, and this is going to hydrolyze the cholesterol and the fatty acid that's on the hydroxyl group on the ring. And then this, of course, can be taken up by the fatty acid transport protein, and this can be, cholesterol be taken up by the NPC1-like 1 transporter. So once we get these into the intestinal cell, then we must construct these into chylomicrons. Because remember that the transport of fats in the bloodstream presents a serious chemistry problem. The fatty acids and cholesterol are not transported by themselves because high concentrations of fatty acids and cholesterol can be toxic to cells. They can be damaging to membranes, so the cells need to be protected. So in order to do that, we're going to construct our chylomicrons. So in order to do that, the intestinal cells will take 2 fatty acyl-CoA groups, add this to the 2 monoglyceride structure, and what we are going to generate here is the same triglyceride structure that we are using for transport. Our 2 coenzyme A's, of course, are released in this reaction, and we generate our triglyceride. The next is to take a fatty acyl-CoA group, add this to cholesterol, and we are going to construct our cholesterol ester structure. So essentially we are taking and breaking these compounds, these structures down, the triglyceride into the fatty acid into monoglyceride structure, cholesterol broken down to cholesterol and plus the fatty acid, the esters, and then we're going to reconstruct these in the intestinal epithelial cells. Now once we have this triglyceride and we have cholesterol esters and we also have free cholesterol, now we have the substrates that are going to be used to construct the chylomicron. The chylomicron structure is going to be assembled on the endoplasmic reticulum surface. So recall that when we do secretion of proteins, in our case we're going to be talking about ApoB48, which is the apolipoprotein that is responsible for the structure, the skeleton of the chylomicron. This is going to be translated into the ER because it is a secreted protein. As this protein is being synthesized, it begins to fold. So the folding of this protein is determined as cotranslational. So that means that as the protein is undergoing biosynthesis, it is being translocated across the endoplasmic reticulum into the lumen, and as it's folding during the translational sequence. Now, as the protein is beginning to fold and construct itself, it is now going to be acquiring triglycerides from triglyceride transfer proteins. It's also going to be adding cholesterol esters. It's going to be adding cholesterol, free cholesterol that is available, and phospholipids. So the ApoB48, consider it like a serpentine structure, like a snake, and this thing is going to be weaving in and out of these fats, and all of these fats will be forming in the core of the structure. On the outside will be primarily phospholipids. And the reason for this is because the core, which is going to be hydrophobic, is going to sequester these hydrophobic fatty acids such as triglyceride and hydrophobic cholesterol and cholesterol esters, is going to sequester them from the aqueous environment of the blood. The phospholipids, on the other hand, are amphipathic, which means they have both characteristics of hydrophilic and hydrophobic. And these will be placed with the hydrophilic head groups on the outside of the chylomicron structure, and the amphipathic, the hydrophobic fatty acid tails will be positioned so that they are facing inward toward the hydrophobic core that is containing the triglyceride and phospholipid structure. And so if we look at the final structure of the chylomicron, we can look at it based on its chemistry. So if this is the core, the center, we are going to have triglycerides, we are going to have cholesterol esters. This is going to be the hydrophobic core. On the outside, we are going to have a layer of primarily phospholipids, and we can also have some free cholesterol. Now, why does the free cholesterol anchor itself more toward the periphery? Well, that's because, remember, we have a polar hydroxyl group that's on the ring of free cholesterol, on the A ring. So now we've established the core of hydrophobic molecules. The periphery has the amphipathic molecules, and now we can interact with water in the chylomicron structure because we have these charged and polar head groups of the phospholipids that can form the hydrogen bonding and ionic bonding with salts as we transport the fats through the bloodstream. Now remember, we also talked about that these chylomicrons are approximately 500 nanometers in size, and so these will enter the lymphatic system in the intestine because they are far too big to enter the capillaries. Capillaries are going to require much smaller particles in order to get through those endothelial junctions in between the endothelial cells that form the capillaries, but the lymphatic endothelial cells are leaky. They have larger pores, larger openings between where the chylomicrons can slip through and then eventually get to the blood. So now that's the chylomicron structure. Now let's look at the structure of the next lipoprotein carrier, and that's VLDL, which stands for very low density lipoprotein. Now the VLDL, let's start out with how it is constructed, and let's start out in the liver in the fed state. And liver in the fed state, recall, is going to have a plentiful supply of glucose coming in by the GLUT2 transporter. And this glucose, remember, will form glucose 6-phosphate. The energy charge of the cell will be maximized by glycolysis. We will be filling our glycogen stores in the fed state, and if we need biosynthetic reducing power in the form of NADPH, we will be shunting some of the glucose 6-phosphate into the pentose phosphate pathway. Now, once our energy charge here, our concentration of ATP has maximized so that the cell is happy. Once that our concentration of glycogen is starting to become full, saturating, then the glucose energy capacity is going to be stored as fatty acids. And so this is where we will end up with our pyruvate at the end of glycolysis. The pyruvate will enter the mitochondria. It will combine with, it'll form acetyl-CoA, which will combine with oxaloacetate, forming citrate. And then the citrate will enter the cytoplasm, converting itself into acetyl-CoA, oxaloacetate. The oxaloacetate will pick up some NADH equivalents, forming malate, and then the malate can be transported back from across the the mitochondrial membrane using the citrate malate transporter. Now the acetyl-CoA is going to be converted to malonyl-CoA because recall that the enzyme acetyl-CoA carboxylase is now under stimulation conditions because we have insulin signaling. And so the malonyl-CoA will be our primary substrate to form fatty acids. Now, as these fatty acids form, which are the C16 palmitate molecules, this is going to combine 3 fatty acids plus glycerol 3-phosphate, and then by our acyltransferase enzymes, which will give us our triglyceride structure. Recall that in the glycolytic pathway, we will be converting DHAP to glycerol 3-phosphate by redox reaction using NADH. And then this glycerol 3-phosphate is represented here in the triglyceride synthesis pathway. So now, once we have our triglyceride, we have one of our major substrates to form VLDL. The other substrate is going to come from cholesterol synthesis. Remember our acetyl-CoA? We will combine in a couple of reactions to form our HMG-CoA, which will form our mevalonate and onto cholesterol synthesis. All of this will happen as long as we have sufficient amounts of glucose coming in the fed state. So this is an anabolic situation, and we are synthesizing our major precursors. Once we have our cholesterol, we now can convert this to a cholesterol ester by adding a fatty acid, CoA, fatty acyl-CoA group, to make our cholesterol ester. And now we have our major substrates in order to construct the APO or the VLDL. Now, to do this, we're going to have a similar situation as in the intestinal epithelial cell, where we're going to need a skeleton protein. So, in this case with the liver, it is going to be synthesizing the protein ApoB100. Just like in the intestinal epithelial cell, this will be translated into the ER lumen, and during this co-translational process, the triglycerides and cholesterol esters plus phospholipids that are being constructed will all combine to form the final VLDL lipoprotein particle. Now recall here that the phospholipids, if you're wondering where those come from, we have glycerol 3-phosphate here that's being used for triglyceride synthesis, but we can also use this for phospholipid synthesis because we're in an anabolic state because of the liver being in a fed state, and we have insulin hormonal signaling to tell the liver that we are in a fed state. Now, once we have the VLDL particle, which is, again, remember, it's about 10% the size of the chylomicron structure, this now can be secreted directly into the blood by the liver. This enters the blood, and now this is going to provide cells with a long-term fat source for energy. Now, I think I also mentioned another level of regulation here to be aware of is that if this pathway of VLDL assembly, it becomes rate limiting, so that the combining of all of these hydrophobic storage molecules with ApoB100 to form the VLDL particle lipoprotein. If that pathway becomes saturated and rate limiting, we have to slow this rate of fatty acid synthesis. And the way we do that is that the fatty acid here, C16, which is palmitate, this is going to feed back and inhibit the acetyl-CoA carboxylase enzyme, which is going to slow the conversion of acetyl-CoA to malonyl-CoA. In doing that, we are reducing the concentration of substrate that is required for fatty acid synthesis, which will slow the fatty acid synthesis down. Okay, now that we've formed our chylomicrons, from dietary fats, and we have formed our VLDL particles from fatty acid synthesis in the liver in a fed state, let's take a look at the pattern of lipid flow in the body on what happens. So our chylomicrons are secreted by our intestinal cells. The VLDL, of course, is secreted by liver cells, hepatocytes. And during the fed state, in the postprandial state, we have our chylomicrons. These will circulate in the blood, and they will provide tissues that need fat. For example, skeletal muscle that will use fatty acids for energy, or adipose tissue for storage of these dietary fats. As the chylomicrons pass this tissues, we'll see the mechanism in the next video, they are going to extract material that they're going to need. And the extraction of material is going to be fatty acids plus some cholesterol. Primarily, though, this is going to be the delivery of fatty acids from the dietary sources to the tissues. Once this happens, we form now what are called chylomicron remnants. These chylomicron remnants, then, are what are considered spent particles, which means that they have done their job from delivering dietary fats from intestinal absorption to the tissues for a period of time, and then these chylomicron remnants will be picked up and internalized by the hepatocytes, the liver cells. The liver will then disassemble the chylomicron structure, and it will reassemble the fatty acids plus cholesterol plus the cholesterol esters with the skeleton protein that it is synthesized, which is the ApoB100. Once this happens, these structures now are secreted into the blood by the liver in the form of VLDL. So the pattern of lipid flow here basically is that the chylomicrons are a short-term supply of fat, fatty acids, the VLDL in the blood is the long-term supply of fatty acids and ultimately also cholesterol. Now, this allows the body to make sure that there is fatty acids available at all times, both in the fed, postprandial period, and in the fasting period through the VLDL. Now, once the VLDL enters the blood system, it is going to be processed just like the chylomicrons were. We are going to be extracting fatty acids as it circulates in the blood. And then with the continued processing, which means the lipase activity we'll see in the next video, to release these fatty acids from VLDL, the particle size will decrease its lipid to protein ratio. Because lipids are being extracted, the protein, recall, is primarily ApoB100. We'll see other accessory proteins later. But the protein concentration is going to roughly stay the same. The lipid concentration is going to be decreasing because we are continually extracting fatty acids from the VLDL into tissues. As this happens, the density increases, going from very low density lipoprotein to low density lipoprotein. And this LDL component here now is very, very much reduced in its fatty acid content, and this will essentially be enriched in cholesterol and cholesterol esters. Now, cells can also take up this LDL, and it will provide them a source of cholesterol. If cells do not pick this up, our friend, the liver, will also remove the LDL from the circulation and disassemble the LDL particle, and in doing so, it will degrade the ApoB100 protein, and the cholesterols that remain, that are being extracted from here, will be eliminated from the body through bile salt synthesis. So the cholesterol is the major substrate for bile synthesis, and then the bile synthesis, once the biles are formed, the bile, recall back when we started talking about dietary fat intake, this will be secreted into the intestine to emulsify new dietary fats for pancreatic lipase activity and fat uptake in the body to reconstruct new chylomicrons. So basically we are using a waste product here to generate the smaller dietary fat lipid droplets to increase the lipase efficiency to release more fatty acids to reconstruct chylomicrons. Now, the other aspect we need to keep in mind is also HDL. HDL stands for high density lipoprotein. And HDL is different from the LDL and the VLDL. It's very easy to make the mistake of saying that the VLDL is processed to remove fatty acids to create LDL, and then the LDL is processed to HDL, but that is incorrect. HDL is a separate particle. It also has a central core protein, like the chylomicrons in VLDL, but it is not considered an ApoB structure. It is called ApoA1. So this is the core protein from HDL. Now the HDL is high density because it has a very, very low lipid to protein ratio. The primary function of HDL is to remove excess cholesterol from cells. So LDL delivers cholesterol to cells. HDL removes excess. So the cell will use as much cholesterol as it needs, and any excess will be removed by HDL. Now, when we talk about a clinical standpoint, this is why when you get blood tests back and your physician orders a lipid panel, they're looking for triglycerides, they're looking for LDL cholesterol, they're looking for total cholesterol, and looking for your HDL levels. So clinically, you are going to want to have reasonably low triglycerides. And the reason for that is because that will indicate that you have a reduced amount of fat in your blood. Excess triglyceride indicates that you have too much chylomicrons, too many chylomicron remnants, and too much VLDL, which means that you have too much fat being taken up into your circulation by dietary sources, or too much fatty acid synthesis by your liver. So your triglyceride levels should be low. Total cholesterol also should be low. But remember, total cholesterol, this is a combination of your LDL content of cholesterol plus your HDL content of cholesterol plus any free cholesterol that may be in your circulation. So the total cholesterol value is not of great importance anymore because we now know that the HDL cholesterol is your good cholesterol because that's your carrier to get rid of it. So we're going to focus on the LDL. So your physician will typically look primarily at the LDL cholesterol. This also should be low. And this is an indication that your liver is taking up excess in your LDL in your circulation. And it's also an indication that your dietary cholesterol is low. Dietary is low, and also your hepatic synthesis is low, which means that you are maintaining a proper cholesterol balance in your body. Now in your lipid panel, the HDL should be high. And what does high mean? As high as you possibly can get it, because this is going to be involved in removal of excess cholesterol. This is the vehicle to remove cholesterol. So if you have low HDL concentrations, that means that you are going to have excess cholesterol. So low HDL means excess cholesterol in tissues. Excess means in excess of what the tissues need. Now picture a low LDL level. Picture your coronary arteries. And do you want excess cholesterol being maintained in your coronary arteries? The answer is a big resounding no, because this is the primary substrate that leads to atherosclerosis, which we'll talk about in a later video. Okay, so the final topic in this particular video series we need to talk about is the purification of the lipoproteins we've been talking about, these lipoprotein carrier proteins, purification and their nomenclature. How did they get these names that we talk about? Well, first of all, let's define their names based on the purification scheme that is used to identify and isolate and identify these lipoproteins. So the first thing we're going to talk about is taking blood from an individual, and we're going to identify this individual's lipoprotein content. We're going to put the blood into a tube. We are going to allow the blood to, in this case, remain as blood. So we are going to take what's called citrated blood because we want to keep the plasma fraction as opposed to generating serum. So we are going to undergo a centrifugation spin. Centrifugation, we're going to spin these tubes, and the first spin is going to be 2,000 times the force of gravity, 2,000 times g, for approximately 15 to 20 minutes. And what we are going to obtain after this spin is a pellet, and these are going to contain our blood cells. This is not what we want. What we typically want in this to isolate the lipoproteins is the plasma fraction. The plasma fraction is going to contain our lipoproteins. So now we are going to next take the plasma fraction out of this tube, and we are going to add it to the next tube. To this tube, we are going to centrifuge at 100,000 times G for approximately 10 to 15 minutes. What we will get here is a fraction on top, and this fraction will be our chylomicrons. The chylomicrons float under these conditions. And why do they float? Because they have very, very low density. And that is determined by the very high lipid to protein ratio in this particle. Recall that these particles have primarily ApoB48 because these come from the intestinal dietary construction, and they have lots of lipid in there because these are dietary fats, and recall that they are 500 nanometers in size. So because of this density of having high lipid to protein ratio, as well as a very large size, during centrifugation, the frictional coefficient is going to be very high because of the size, and the density being low, it's going to be pushed up to the top of the tube. So in order to isolate our chylomicrons, all we have to do is to pipette off this top fraction. Now, the remaining plasma material is going to contain our VLDL, our LDL, and our HDL. So in order to separate these, we are going to put these onto another tube. But in this case, we are going to mix this material with a reagent called OptiPrep. And OptiPrep generates what's called a self-forming gradient. And what that means is upon centrifugation, which at this time is going to be increased to approximately 350,000 times g, and we are going to do this for a minimum of 3 hours. That means that the density in this tube is going to be a gradient with low density at the top, high density at the bottom, and this density is determined by the OptiPrep. So the particles in the OptiPrep under centrifugation pressures, the lower density particles will move to the top, the higher density particles will move to the bottom, and we will have a continuous gradient of densities. What that allows us to do now is when we have our plasma mixed in here, the lipoprotein particles of interest are going to migrate in the tube to their correct densities. So near the top you can predict that the VLDL particle will be near the top. Just below that we will have a band of lower density particles, which is LDL, and then closer to the bottom we will have another band of lipoprotein material, which will be defined as HDL, high density lipoprotein. So how does this actually work? In the beginning, these are all mixed together, and as the centrifugation continues, the particles themselves will move back and forth in the solution. So for example, if we start out with a VLDL particle here, based on the density, it is going to be pushed up in the tube. Even though the centripetal force is going out, it'll be pushed up into the tube at its correct density, and it will remain there. Now the nice thing about this self-forming gradient is this time is a minimum of 3 hours to form the gradient, but you can technically spin this, centrifuge this sample for indefinitely, and the gradient will not change. So this is how our densities are determined, and this is how the names of these lipoproteins were assigned based on the densities. And the densities are primarily determined by the lipid to protein ratio and also the size of the particle. Between these two will determine the migration position of each one of these particles.